In this video, I'm going to show you how to run an Agarose Gel Electrophoresis in order to separate out your PCR products of the 16S rRNA gene from your isolated colonies. Agarose Gel Electrophoresis takes advantage of the fact that DNA has a negative charge. We will apply a current, and so the DNA will move from the negative charge towards the positive charge, from the negative pole towards the positive pole. And we have the DNA move through a matrix made of a compound called agarose. And what happens when we make an agarose gel is that there are pores within that gel. They're of course microscopic, and the DNA fragments move through those pores. Smaller fragments can move faster, larger fragments move slower. So what we would see if we had multiple fragment sizes is the shortest fragments would be towards the bottom of the gel, the larger fragments would be towards the top. Making a gel is a relatively simple process. We have to decide what percentage agarose we want to use. For our fragments, I'm going to use a 0.8% gel. And so what that means is that for every 50 milliliters of gel, I will need 0.4 grams of agarose. So I'm going to weigh out 0.4 grams of agarose. I'm going to measure 50 milliliters of buffer. It's called TAE. So it's tris, it's acetic acid, and EDTA. And then I will microwave that, and we will add a visualization agent, and then we will pour our gel. Okay. And we'll weigh out the 4 grams of agarose, 0.4 grams. Easy enough. And then we will add that to a flask. And so I have a dedicated flask in my lab for doing Agarose Gel Electrophoresis. So I'm going to pour the powder into the flask. And then I'm going to measure out 50 milliliters of the TAE buffer. So 50 milliliters, add that to the powder, give it a shake, and then I'm going to put this in the microwave. Now the thing with agarose and TAE is that it gets a lot hotter than water, so I have to be very careful not to burn myself when I'm doing this. So it'll boil over really quickly too. So what I do in this case is start at only 25 seconds, and then I watch to make sure that it's not bubbling over. If it starts to bubble over, I turn off the microwave, let it calm down, and then add a little bit more. So I'm going to add more time, but in descending quantities. So I added 25 seconds. It hasn't boiled over. So I'm going to add 15 and do it again. So this will be a process that I'll go through until the agarose TAE solution looks clear and doesn't have any bubbles. I'll show you what it looks like after this step, and then I'll pause the video until I get it clear. So what we see right now is a lot of bubbles and kind of some chunky stuff. We don't want that. So I'm going to go ahead and continue to microwave until it gets clear, and I'll show you that in just a few minutes. Okay, so hopefully you can see now how clear it is. This means that once it cools down a little bit, because it's still relatively hot, I'll be able to pour the gel. So I'm going to pause the video to let this cool down, not too much, because when it cools down it's going to re-solidify, but to cool down just enough so that I can add a chemical called ethidium bromide. Okay, so I have gone ahead and let the agarose cool. So I'm going to add some ethidium bromide. See here, ethidium bromide. This has the potential to be a carcinogen. So what I'm going to do is very carefully take out 2 microliters of this solution, and I'm going to mix it with my Agarose TAE mixture. I'm going to swirl that around so that it mixes carefully. And then I need to actually change gloves because I don't want to touch my camera and move it with gloves that might be contaminated. Because this is a chemical waste, I'm going to put my gloves in the chemical waste container. So I'll be right back. Let me get some gloves. All right, fresh gloves. And so now what we're gonna do is actually pour the gel. So what we have here is a gel casting system. We'll pour the gel into this device, but we don't want it to leak. So we'll tighten this up. And that makes a really nice seal. And then we have to have a spot to run each individual sample. So we use these combs shown here. So I'll put one comb at the top. There are a lot of samples. One comb at the bottom. Each of these combs will allow me to run 15 different samples. So for one sample, I'll have to run a ladder so we can know that our fragments are the appropriate size. And then in the other wells, I'll run your samples. So next, what I'll do is simply pour my agarose solution into the gel mold. There may be some bubbles. If there are bubbles when you pour a gel like this, you can always use a tip, like a pipette tip, to pop the bubbles, but then make sure that that pipette tip goes into the chemical waste because it will be contaminated with lithium bromide. So we'll wait for this to solidify and then I'll show you how to load and run the gel. So the gel has solidified. Now I will take it out and set it up in the device so that we can load our samples. So the first thing we'll do is remove our combs. Then this is the system where we run our gel. And so we'll open this up and bring it a little bit closer. Undo the casting system. Pull out the gel and put it in the tray. There's not quite enough buffer, so I'm going to fill it up with buffer and then I'll show you how to load a gel. Okay, so I've covered the surface of the gel with more buffer. You want to make sure that buffer covers everything. And the first thing I'm going to load is in the first well of each row, I'm going to load what's called a DNA ladder. The DNA ladder contains fragments of known sizes that will separate out when we apply the charge. And so these tips, because they are going to touch the gel, end up being contaminated with lithium bromide, and so we discard them in the chemical waste. So what I'm going to do now is load 5 microliters of each of your samples into these wells. I won't make you watch the whole thing because it does take some time, but I'll let you watch a few of them. And so you may be asking yourself, why do our samples not float out into the liquid? Because it's a bunch of liquid and you would think that it would float out. But what this PCR master mix has in it is a little bit of glycerol. And so glycerol weighs those samples down and forces them to kind of sit in that well. Without the glycerol, our samples would most definitely float away. So again, I won't make you watch me load the whole gel. I'm going to go ahead and pause the video and then I'll turn it back on when we actually start running the gel. Finish loading the gel. You can see the green representing your samples in the wells, except for over here I had more wells than I had samples. So now what we'll do is go ahead and get this set up to run. So we take the lid and we're going to match the black port to the black port and the red port to the red port. The black port is the negative charge, the red port is the positive charge. And remember, DNA is negatively charged, so it should be repelled by the negative end, and it should be attracted to the positive end. And the DNA will move faster if it's small and slower if it's a large fragment. So we have our power supply. Let's turn that on. Okay. And we will set it to run at, let's do 100 volts. And then we'll change to 20 minutes. So run. And what we'll see when it starts running, you see the bubbles coming up over here and over here. And so that means it's running, and the arrow tells us in which direction our fragments should be moving. So we'll come back in about 20 minutes and check on that gel. Okay, so our Agarose Gel Electrophoresis is done. We can see that it has stopped running. So what we'll do now is remove the gel from here. I'm doing this one-handed, so unfortunately it's a little bit tricky. Okay, I will grab the gel, and you'll notice I'm wearing gloves because this is contaminated with ethidium bromide, and we want to do the best we can to keep ourselves safe. And now we go over to, this is it's called a trans illuminator. It basically shines UV light. And the reason that we are going to shine UV light is because that ethidium bromide intercalates, or gets in between the bases of DNA, and it also is UV light reflective. So what should happen is that wherever the DNA is, we should see a band pop up when I turn on the UV light. So let's see what happens. Okay, so we see some bands. And what I'll do is, the video quality isn't great. I will take a picture of this gel and add it to the end of the video and kind of talk you through what it is we are seeing. So if we look at this gel picture, in the first well of each row, you'll see multiple bands. This represents the ladder. In the ladder, these are all fragments of known size. It's a little bit hard to see, but you have 100, 200, 300, 400, and then you have this thicker band. That's 500. And then you have another thicker band that is above that, and so that one is 1,000, and then above that you have, I believe it's 2,000, that is the other thick band. The fragment size we're looking for is anywhere from 1,000 to about 1,250. And so unfortunately, with this first round of PCRs, it doesn't look like too many people got amplification of the appropriate size. I will add a little arrow to the video, and you can see which had bands of the appropriate size. But it is my understanding that you also did the PCR a second time. So I will be running those out when the shelter-in-place order is over. But as of right now, I've only had time to run the first set. But again, hopefully what you understand is that near the top in a lot of the wells, you'll see this really bright band. That just is the total DNA that was in your cells. Towards the very bottom of each set of rows, you see a band that's lit up down there. That is what we call primer dimers. Sometimes the PCR primers bind to each other and amplify themselves, and that's where you get primer dimers. But then pointed out by the arrow are going to be bands of the appropriate size. So you have the really small bands of the primer dimers at the bottom, our appropriate size somewhere in the middle, and then near the very top is the total genomic DNA, which is of course very large and could not make it all the way through the gel during the 20 minutes that we were running.